Great, thank you very much. So we had a very useful discussion, actually. It was very good. Contributions from everybody there. The challenges we were specified at the start were how can we develop regulatory pathways, what roadmap would we use, how can we make them dynamic, and how we make them acceptable within the NHS to patients, industry partners and overseas. Our conversation, I will be honest, steered somewhat away from that and started with more generalised worries about things that have been brought up earlier on, and we spent a bit of time at the start discussing the the healthcare exemption work and what constituted healthcare bodies or collaborations. And we ended up deciding that was very complicated and very complex, because actually the whole process is very complex, and what you've got is lots and lots of different parts. Who makes the gel? Who makes the molecules and the chemicals that you're using? Who produces the reports? Who synthesizes the reports for somebody else? Who interprets the reports? And who gets sued when it all goes wrong? And we decided that was actually quite a complex process and probably needed more thought than just ourselves. With regard then to the main meat of our discussion around the genetic pathways, we sort of started with the patient and worked outwards from there. So the first thoughts were how you interact with patients and the consent and the documentation that goes with it. So we were concerned about making sure there was very universal, clear consent, clear rules about who accesses the data, clear rules about what the data is going to be used for, clear rules about data sharing, both at a personal family level and at a data cloud, big data NHS level, and concerns about how people would access the information at the right level for them. Some people want to know very little, some people want to know everything, different members of different families will want to know different amounts. Some family members will not want to be involved. How do you tailor and signpost people to the correct level of information for them? How do you account for different levels of educational achievement? How do you account for different languages? How do you account for different accessibility? How do you account for a different doctor sitting in front of you talking to you because the person you're feeding back to will be different? So there was a lot of patient-based discussion. We then talked about the sample pathway once you've had your genetic test taken. So the thoughts there were, it was acknowledged there are clearly differences at the moment in how samples would be handled for cancer samples, and these are not going to be ironed out by the 1st of October deadline, but we could see that if you had the right enablers and the right sticks, then actually this could be driven forward and made better. And the examples that were given were the formalin, removal of formalin from St George's Theatres. That means that you cannot ruin samples, so that enables the test to be done. And then there's sticks. The best example we could think of off the top of our head was not being able to prescribe particular drugs unless you have the genetic information in front of you. So those two factors. We then talked about the informatics systems that were being used to deliver these. Can they be made to work properly? How do we account for the technical aspects of this? Do we have enough staff to deliver it? Do we have the right systems for keeping them updated and up to date? What happens when the next system comes along or the next bit of information comes along? How's it integrated in? We wanted to look about the evidence for the test themselves, and is there evidence of cost benefit? And one interesting aspect that's brought up is not just cost benefit of, if you do this test, can you make sure the right number of people are directed to the right pathway, but what are the knock-on effects? Will you need more physiotherapists? Will you need to use more drugs from a particular drug budget? And the siloing of drug budgets was brought up, siloing of costs and how that's going to be linked together. The next step of our discussion was around how industry could be brought into this, and were there any existential objections to linking in industry to start with? And there weren't, actually. It seemed quite agreed that in the right circumstances, then actually partnering with industry to achieve patient benefit was very appropriate. It's just being careful to work out how you define patient benefit and where you would start to say this or isn't beneficial, and where you draw those lines requires more than half an hour in a committee room. But the principle was actually uncontroversial in our particular group. We then discussed the role of a clinical geneticist, and that was considered a work in progress, but there were various aspects that need to be clarified. Their role with respect to variant interpretation, education, care modelling, and their role as driving innovation, making sure that we're keeping on the cutting edge of these technologies, because if they're not updated, there's really no point going forwards. Education was a theme that ran through a lot of our discussions, and there was education of clinicians, education of the NHS, education of individual patients, but then there's education beyond that. How do you educate families, children, grandparents of people who've got genetic tests because the information may be relevant to them? How do you educate children in schools so that their level of knowledge increases and therefore the acceptability of these genetic pathways improves? The example was used of a donor card, whereas when they came out, they were very novel and people didn't necessarily understand quite what they were for, and now they are. We don't go around educating about donor cards. People know what donor cards are, they know what the purpose is, and that I thought was a nice analogy perhaps for this sort of test. There was also then a question of different quality management systems in terms of looking after genetic tests, as different systems will prioritise different aspects of the data quality. So they may provide stronger risk assessment or provide different areas of other information, and this, depending, as there's no agreed standard, if different people are using different systems, you will drive different tests in different directions, and the updates and the incremental improvements of these tests, you're nodding, I'm getting this right, aren't I? Good. Will therefore, you'll get variation, you'll get divergence over time, just by applying different quality management systems. So even details as minute as that need to be considered. And finally, going back to the patient having been all the way through the test, we then wondered how we could deliver this in a real world environment. And there are conflicting arguments here, and I don't think we came to a satisfactory conclusion. On the one hand, can we empower people to collect information? Because that would reduce the workload implications massively, but it would potentially skew health inequalities because it's going to be more easily done by people who have good education and high health-seeking behaviours. They're going to input the data onto a system to say how many relations have had breast cancer, and they'll know how many relations have had breast cancer or uterine cancer, whereas a 15-year-old in care might not know any of those things and might be therefore disadvantaged. On the other hand, a busy clinician with 15, 20 patients in their clinic is going to find it very difficult to collect the depth of phenotype needed to interpret these tests properly in the current setup. So we've either got a disruptive technology to current systems, or we've got to think about how we incorporate the data collection in a way that is as equitable as possible across different groups.
Great. So we had a very diverse group, had a very interesting discussion. We did diverge a bit from the questions that Munir had given us. I hope he'll forgive us. But no, it's too difficult. Actually, no, we did first of all discuss the roadmap that we decided was actually more of a road cycle, if that makes sense, because we thought the easy bit was probably getting to the CE mark, easy-ish, but beyond that, you've got all these other organisations and factors to consider. So you've got NICE, you've got UKGTN, you've got commissioning process to agree, and then you've got uptake, and related to uptake is training and perhaps reminders on records that we've already discussed. And then you've got vigilance. So there's the vigilance of the medicine, perhaps for a companion diagnostic, but there's also the vigilance for the device, and that's different. But we feel that sometimes they need to be considered together. So the vigilance for the device might be false positives, false negatives, how you would detect that, and whether you would report it. But also, what if the test actually isn't used and it should have been used? Is that something that should somehow get captured and reported? So what we're going to do with the road cycle is Steve is going to draw it. And then we, as a group, we'll perhaps...
On the back of Munir's question sheet.
And then we'll agree as a group and then let you have a look at it, Munir, for the report. So we spent a lot of the time talking about performance evaluations. So we said that they need to be fit for purpose. So the minimum amount of regulation and evidence, if you like, that would be fit for purpose. So the regulation needs to be proportionate. It needs to take account of risk. It's also a fast-moving field. So as soon as we've actually done the performance evaluation, the chances are it's no longer valid because things have moved on. So that was a concern. We compared the new regulation with the old directive, and we noted that we now need to include scientific validity and clinical performance. And I always thought as a clinician that the clinical performance bit was the most difficult, but actually we spent a lot of time talking about scientific validity. And some people in the group wondered if, particularly for genomics, whether scientific validity and clinical performance could actually be the same thing. And would the scientific validity be enough, perhaps with some sort of bridging? We feel that we need guidance for the scientific validity. So one suggestion was that the UK GTN dossiers would actually provide very good evidence of scientific validity, and perhaps this could be taken forwards. There was a discussion about rare disease. So it was noted that for medicines regulation, we have possibility of approving drugs in exceptional circumstances for rare diseases. So there wouldn't be very much data. There might not be any comparative data. So this isn't specifically in the IVD regulation, but Steve pointed out that it says you need sufficient clinical evidence. So it may be that for a very rare disease, it might be acceptable to have just a handful of patients. So we went on to explore what a CE mark genetic test might look like. And our industry stakeholders in the group talked about the Fda approach, and that's where you actually have components of the test that are CE marked in their own right, because a lot of the components are going to be the same for tests that are looking at different variants. So then it's a sort of like a jigsaw or a modular approach that could be used. And when it all fits together, you get something which is sort of validated end to end. There was some discussion about the uncertainty of sequencing in certain parts of the genome. I didn't understand that very well, but my understanding is that that can affect sensitivity, specificity, and it is worse in certain bits of the genome. And this is part of the analytical performance. And should that uncertainty be communicated to the requesting clinician or the patient? And I think the feeling is that that information would be there in the label somehow, but whether it would actually be in the report that was giving advice on, the variant that had been found and its clinical significance, I think we didn't really conclude on that. So we concluded that clinical performance is really more about what does the variant mean for the patient, whereas the analytical performance is can the variant be detected properly. We talked about the role of standards and that these are needed for genetic tests, and whether CLSI standards might be appropriate, and apparently that is controversial. We talked about reference DNA and the fact that this is sometimes important, particularly for certain difficult matrices. We talked about the fact that we decided that performance studies for genomic tests in most cases are likely to be retrospective, perhaps using the biobank, using archived samples and electronic health records. And then there was concern raised that for developers that are essentially piggybacking or developing Me Too diagnostics, that they would need to demonstrate equivalence, but they wouldn't need as much data as the first diagnostic. And in fact, there isn't really a market advantage to being the first, because unlike medicines, there isn't data exclusivity. Obviously, in some cases, a patent might be applicable, but that's something sort of outside of our remit. And the comment was that perhaps at the moment, everybody is waiting for everyone else to jump first. Thank you.
Just put that in a pocket. Perfect. Hello, everyone. How are you all doing? So I'm Mark Bartlett, and I was the rapporteur for Group 3, where we had quite a lot of lively discussions, I'm sure you all had in your own groups. We did venture off topic quite a few times, but you know, that's kind of part of the fun, isn't it? So we had a varied group. We've had people from the NHS, also had sequencing companies, pharma companies, MHRA, academia, even trade bodies represented within the group that we had. I'm going to try and summarise as quickly as possible some of the main themes that kept coming up, which we felt addressed the 5 questions that we were set to task in slightly different ways. So the 3 main points that we felt regulatory we need to think about when talking about regulations is established, exceptionalism, and patience. So we have established frameworks. There are pieces that exist from different areas, and should we be reinventing the wheel? Maybe not. There's the notion of exceptionalism around genetic data, and we feel that actually this conversation should be healthcare data is exceptional. Genomic data is just one part of that healthcare data. So yes, it is exceptional in a way, but not anything unique to what we're already collecting within healthcare systems. And then patient. Obviously, we need to put the patient at the heart of everything. And how do we actually create frameworks that can include patients in a robust, communicated, transparent way? Going to the established. So when I was saying we have established frameworks, we felt that it was quite interesting to investigate breaking down the journey of going from sample to insight or diagnostic into many different steps. Joanne had an anecdote that actually from Genomics England's perspective, there's about 400 different steps with different processes that you need to go through to actually go from sample to insight. So breaking that down into different stages, you can start pulling in the different frameworks that already exist to regulate those different steps of the puzzle. And if you quite broadly branch them into, there's the sequencing element, then there's analysis, which is bioinformatics kind of elements, and then data storage. When we can start thinking of those 3 specific areas, you can start thinking of sequencing. We've got diagnostic or in vitro diagnostic frameworks from the MHRA. Analysis, there's the kite marking schemes or digital analysis frameworks that you can use from NHS Digital. And then storage, we're looking at like GDPR, how do you involve patients in this journey and be in consent, those models there. Another concept that came around was around the life cycle of the different products that are available in those different stages. It's probably too heavy-handed to say that there's just one way of doing this. So you could probably learn from the regulations in pharma, where you have or an early, middle, or late stage application of your technology. So we felt overarching regulation could be light touch within the early stages, maybe a bit more robust in the middle stage, and then light touch in the late stage. So if you think of the phases in the pharmaceuticals going from phase 1 to phase 4, when you're out in the late stage, you'd be in the pharmacovigilance or perhaps the exemption kind of area that the MHRA is proposing earlier. So those were the main themes that kind of came out. The reason that we think about the light touches and more rigorous touches is we're not saying scale back any of the regulation that's already in place. We just want to maintain the ability to move fast and be quick in this whole area. So we can go early stage, bit lighter, and then robust and light. And as I was saying, patients always at the heart. When you're thinking of like data banks and data storage, you think of GDPR. Making it more transparent, getting consent processes in place with patients is really vital, and that always needs to be at the core of any regulation that's going to be acting in that field. So that's a summary.